Prokaryotes tend to regulate genes through repressors, while eukaryotes tend to regulate genes through activators. Eukaryotic activators have a DNA binding domain and an activation domain. The activation domain has a great deal of variety, and so does the DNA binding domain. The two can mix and match. Why do we need activators? They attract RNA polymerase II, the one that transcribes mRNA, general transcription factors, 5 of them, TF2B, TF2D, TF2E, TF2F, and TF2H, and the mediator mediates between RNA polymerase and the activator. Here's an easy way to remember the general transcription factors, by the way. Also, remember that prokaryotes only have sigma factor as a transcription initiation factor helping RNA polymerase bind to gene promoters.
Hey, you forgot to mention another thing we fine fellows have that you guys don't. Us prokaryotes have operons. That means we can transcribe an mRNA with a whole series of genes, and then we just need one promoter for the lot.
Anyway, activating proteins can do the above either directly or indirectly. How? They can modify chromatin structure and increase promoter accessibility. So, how does approximately 2 meters of DNA fit in a human cell? You can thank chromatin for that. Chromatin, the miracle that allows for on-demand access to any particular DNA segment, pronto. The DNA in interphase chromatin is condensed 500 times by length. During mitosis, it's condensed so much you can see structures called chromosomes. Chromatin is defined as DNA bound to histone and non-histone proteins. DNA combines with structural elements called histone proteins to form nucleosomes, which are the fundamental structural units of chromatin. Non-histone proteins are all the proteins that associate with DNA in chromatin minus the histones. Side note, the side groups of the amino acids of regulatory histone proteins hydrogen bond with tightly wound DNA upon recognition of base pairs exposed in the major groove along its backbone. These regulatory proteins share several structural motifs, including the helix-turn helix, leucine zipper, and zinc finger. Histones are relatively small proteins, ranging in size from 102 to 135 amino acids, with a strong positive charge due to the basic amino acids lysine and arginine making up over a fifth of their amino acids. This allows for electrostatic attraction, 142 hydrogen bonds, to negatively charged DNA, and causes the DNA to be negatively supercoiled in such a way that it can be unwound locally. When bound to the positively charged histones, DNA's negative charge is essentially neutralized. Hydrophobic interactions and salt linkages also contribute to keep the histones and DNA together. There are 5 major types of histones: H1, H2A, H2B, H3, and H4. The last 4, that is, all the histone proteins excluding H1, are called core histones because they form a histone core, which is an octamer consisting of 2 of each kind of histone protein. H1 is termed a linker histone, as it associates with linker DNA between histones. It is found at the location where the DNA enters and leaves the nucleosome and stabilizes this region. A nucleosome plus 1 bound H1 protein is termed a chromatosome. Loosening or removing this H1 protein can allow a nucleosome to slide along DNA and thus grant access to transcribing enzymes. Histones combine with DNA in approximately 1-to-1 ratios by mass. Nucleosomes not only help DNA get packed into the nucleus, but also provide a great deal of protection against mutations to the 147 base pairs of DNA wound about 1.7 times around the histone octamer. Note that the 147 base pairs are only the length of DNA wrapped around the octameric histone core. The length of DNA between nucleosomes is called linker DNA, and it varies in length from about 10 to 80 or 90 base pairs. This pattern of nucleosome linker is sometimes called beads on a string, and the thickness of this fiber is about 10 nanometers, whereas DNA helix is about 2 nanometers thick.
Note that although folks will often refer to a nucleosome as just the histone octamer wrapped 1.7 times by DNA, the technical definition of a nucleosome is the wrapped histone octamer plus 1 adjacent length of linker DNA. Hence, you will find about 1 nucleosome per 200 base pairs.
Regardless of sequence, DNA can almost always successfully wrap around the histone core, though it might have to take a slightly crooked path. In general, nucleosomes orient themselves along DNA at random, and alignment is lost when the gene becomes active. However, Sometimes the nucleotide sequence does influence where nucleosomes are located, and there is preference to certain locations. This fairly rare phenomenon is called nucleosome phasing. Nucleosome phasing mostly occurs due to preferential bending of the DNA in locations where AT base pairs are at the bend, as the AT base pair rich areas narrow the surface grooves more easily than GC base pair rich areas do. GC base pair rich areas preferentially occur on the outside of the bend. Always keep in mind that the AT conjugate pair has 2 hydrogen bonds, while the GC pair has 3, and is therefore stronger. The nucleosome positioning preference dictated by nucleosome phasing is weak enough to allow an overpowering of the preference by other factors. Otherwise, you wouldn't get transcription of these DNA regions. That being said, the main factor affecting position of nucleosomes is proteins tightly bound to the DNA. Imagine the nucleosome is the small black car trying to park, and you have a massive truck, a protein, taking up 1.5 parking spots. That car isn't going to park next to the truck because there's no room.
Get out of my parking spot!
Right away, sir. Right away, sir.
Okay, well, that wasn't supposed to happen, but, in the usual situation. The nucleosomes are packed into chromatin fibers, and there are currently 2 theories on how this occurs, the zigzag model and the solenoid model. It is likely that a combination of both packing methods is employed. This compacting of nucleosome beads on a string is termed a higher order structure, and it is approximately 30 nanometers in thickness. Beyond the winding of nucleosomes into chromatin fibers, there are several more levels of winding due to scaffold proteins. I won't describe that in this video, until you finally get a chromosome made of 2 identical chromatids, each about 700 nanometers thick, and humans have 46 chromosomes. This tightly packed mass of DNA and histone proteins is termed heterochromatin, and it is transcriptionally inactive. Genes with heterochromatin are termed to be silenced. Euchromatin, on the other hand, is the extended open form of chromatin, and this form is transcriptionally active. Remember those activator proteins I mentioned at the start of the video? Well, activator proteins can alter chromatin in a few major ways. DNA doesn't just stay firmly wrapped around a nucleosome at all times unless bound to transcription and replication machinery. Other proteins can bind the DNA, because about 4 times per second, the DNA uncoils from the histone for 10 to 50 milliseconds at a time. However, this uncoiling of the DNA is insufficient for many processes, and so we have ATP-dependent chromatin remodeling complexes, of which there are dozens of different kinds, called in by activator proteins. These proteins bind both the core histones and the DNA, and, over a bunch of repeated cycles of catalysis, use the power of ATP hydrolysis to slide the histone cores relative to one another. In the process, the nucleosome core's structure is momentarily modified, loosening its grip on the DNA.
Just a side note, they need energy because they're breaking electrostatic bonds between the histones and the DNA.
Histone chaperones are negatively charged proteins that can work with some remodeling complexes to remove or exchange the 8 core histones for a nucleosome, or replace H2A, H2B dimers with other variants.
All of this was done in my presence.
In order to understand covalent histone modifications, we first need to look more closely at the structure of the core histones, for example H2A. Histones have globular nonpolar domains, as well as basic domains that extend to one or both opposite ends of the histone. The nonpolar domains tend to have conserved sequences between species, while the extended, charged regions are variable. Although histones are superconserved in structure, cells have small numbers of specialized core histone variants. Anyway, a structural motif, called the histone fold, is shared by the 4 core histones. This motif consists of 3 alpha helices joined by 2 loops. When nucleosomes assemble, these structural motifs bind together, H2A with H2B and H3 with H4. The two H3-H4 pairs then combine, forming a tetramer. The tetramer then combines with two pairs of H2A, H2Bs, thus forming the octamer. One of the extended ends of the histone protein is an N-terminal tail, and these tails allow a way to unwind DNA from around the histone cores via reversible covalent modifications. In these tails, lysines can be acetylated or methylated, while serines can be phosphorylated. Side note, addition of a phosphate group is done by a kinase, an acetyl group, acetyl-transferase, and finally, addition of a methyl group is done by a methyl-transferase. In active DNA, histones tend to be more highly acetylated, for this acetylation decreases the electrostatic attraction between the DNA and the histones. If acetylation causes H1's removal, then the DNA is especially easy to unwind. Also, note that the globular regions of histones can also be covalently modified, but I'm not going to discuss that in this video. Modifications to histone tails result in a histone code, which can signal anything from
"Hey, this DNA is damaged,"
to
"This is when gene expression should take place over here."
Histone codes are written by writers and read by readers. Writers are histone-modifying enzymes, and readers attract additional proteins to the chromatin for the performance of various biological functions. Code reader complexes are composed of a scaffold protein with some protein modules. These protein modules recognize and bind to specific histone modifications, and if enough protein modules in a code reader complex recognize protein modifications, then the code reader complex can bind tightly on. The code reader then acts as a middleman to allow other proteins to attach to it and carry out biological functions. There's another cool trick that can be done with chromatin modifications. They can spread along a chromosome over long distances. This is useful to condense or decondense chromatin and can occur thanks to large protein complexes with many of both writer and reader proteins. Basically, a chain reaction can occur for some modifications. First, a gene regulatory protein calls in a writer. After the writer does its work, a code reader binds tightly to the newly modified histone. This reader plus the writer make up a reader-writer complex. This reader will then recruit another writer protein, and so on and so forth. As a wrap-up for this video, I'd like to provide an example of how to write a histone code for transcription initiation. Note that the following steps can only occur in the order presented. Here's a histone core with an N-terminal histone tail coming off each histone protein. Here is the TATA box, which is in the promoter and usually 25 to 35 base pairs upstream of the transcription start site. Here is the binding site for the activator protein. To start the process, an activator protein attracts HAT, histone acetyl-transferase. HAT acetylates lysine 9 of histone H3 and lysine 8 of histone H4. Next, the activator protein attracts a histone kinase, which phosphorylates serine 10 of histone H3. Lastly, the acetyl-transferase responds to the serine phosphorylation by acetylating lysine 14 of histone H3. This completed histone code allows for the binding of TF2D and a chromatin remodeling complex to the histone tails and initiates transcription.